Thank you very much to all the folks at CDF for the kind invitation to come and talk to you today. They're a great group and very resourceful, and please reach out to them whenever possible because they're always willing to help. Thanks, Janelle, for a great talk, very informative. And again, I'm going to refer a little bit to some of the things that you've said in my talk. I'm here as a celiac patient and a researcher, and I really reinvented myself as a food scientist after I was diagnosed myself with celiac disease in the early 2000s. After many years, as you all have, of misdiagnosis and frustration, being fortunate enough to have a laboratory at my disposal, I kind of questioned the gluten-freeness of some of these gluten-free foods when I was first diagnosed. I was trying to learn what I could eat and what I couldn't eat, and did a lot of analysis myself, and found cases where cross-contamination and mislabeling occurred, and found there are occasionally circumstances where you do get inadvertent gluten in some of the food that you buy, as well as the restaurants you go to. And so that's how we founded Immunogenx originally, as to bring some more sensitive and comprehensive methods of analysis for gluten to the market. And since then, we're still doing that, but we're also developing a therapy and a diagnostic to help people manage the disease after diagnosis. And so I'm going to talk today about the food analysis part of our company. And I'm going to talk a little bit about food analysis, a little bit about the analytical methods that are currently used to certify gluten-free foods and to study food in general for allergens. I'm going to give some, sample analysis, some of the not infrequent cases of food that I have seen be gluten-free. Not to scare you, but just to raise your awareness, to make sure you read the labels, as Janelle pointed out. And also, I'd just like to talk a bit about our research and how it's moving forward in the food analysis. So food basically is composed of several different components, but the allergenic component is protein. Food is an exceptionally complex matrix from an analytical perspective. Most people think that clinical samples, i.e. serum and tissue, are the most difficult samples to analyze. Food is incredibly complicated. And you might think that analyzing chocolate or wheat flour is easy. It's not. And so methods that are put out there to analyze manufactured products and native products, there is no one, take-home message is there is no one preferred method of analysis. So it's the food proteins that cause people problems, the physiological responses, allergies, intolerances, immune responses. And so really, the dose is the poison. How much is going to make you sick? And that's different with everybody. And so from an analytical perspective, we always want to see how low can you go. We can detect, although everybody wants to detect lower and lower amounts of targets, allergens, and just because we can. And so people raise the question, well, how low do you want to go? And again, it's really dealing with trying to make our labeling philosophies work for the benefit of everybody. We want to make sure that things that are labeled gluten-free are really gluten-free. So what is gluten? Most people think gluten is one protein. Gluten is not one protein. Gluten is a superfamily of hundreds of proteins. There's storage proteins found in the endosperm of wheat, barley, and rye. they are related, but they're distinct. And here's an example of wheat gluten proteins. They're in two major groups, monomers and polymers, gliadins and glutenins. I'm sure you've heard those terms. And they can be further subdivided and also into several subgroups. And so what are the challenges of gluten analysis? One major problem is that we don't know the identity of all the gluten proteins that are out there. There are a lot of really great researchers. They are constantly identifying new proteins, new gluten proteins, and putting them in databases that we all use to verify that the proteins and peptides that we're seeing in our analysis are really part of those particular grains and not sourced from other types of plants or other species. And so detecting and quantifying trace levels of these proteins is really hard. As you well know, legislation now requires gluten-free food manufacturers to test their foods. And for the most part, they're doing a really good job. It's the situations with the advisory statements, as we heard, that makes things complicated, because no one really knows if they've been tested or not. And so it's really required now that analytical methods be comprehensive to measure just more than one protein per allergen. And because it's so easy to inadvertently contaminate food during the manufacturing and transportation process, it's really a mandated job now to get better, more efficient types of analytical methods to look for gluten. And so I published this paper in C&E News in 2012, and I really started working with allergens diligently around 2010, and this was kind of the beginning of the whole allergen wave in the analytical community. And so really the only method that is predominated and is still accredited by FDA as the enzyme-linked immunosorbent assay, the big mouthful. There are really three current technologies: ELISA, PCR, polymerase chain reaction, and mass spectrometry that are in the playing field right now. And as I mentioned before, there really is no best analytical method. These three methods are the most common. Mass spectrometry is the field that I'm interested in and I have been working with. And so I'm going to probably side towards the mass spectrometry side of things in this talk today. But really, they're complementary methods. There are issues. Everyone has advantages and disadvantages, cases where they work and don't work. And so this is why cases that food have been recalled for a reason because the ELISA didn't work, mass spectrometry can come in and help solve the problem. So I'm just going to go over the background quickly of those three methods. What is an ELISA? It's an antibody-based method, and it has its advantages and disadvantages. It's very specific and very sensitive. However, there are disadvantages in some cases where the ELISA doesn't work, and the ELISAs are what are used for gluten-free certification, because they're the FDA-approved method for analysis. And so the cases where they don't work, foods are recalled or questioned and so on. Some of them have come to me to help in those cases. And part of why ELISAs fail is because of the inherent way that it works. And there are kits available that are not multiplexed assays. They don't look at more than one target protein, and so sometimes there are false positive results, sometimes there are false negative results, and so some fairly hefty assumptions are made with this type of assay, and I'm going to be throwing some science out there at you, and I don't want to bog you down with that, but I'm going to put up some sample analysis traces and so on. So don't get bogged down with that. Just have an overview to the math that I might be throwing at you. Okay, PCR. PCR is a DNA-based test, and PCR is works very well. It's fast. It's very common and expensive. It's had a big history in diagnostics, forensics, as far as recalls, food recalled for salmonella, listeria, things like that. It's used, but it's also being used now in allergen analysis. And so, but it only works if your sample has DNA. And so there are some cases where that's not the case, and also some cases where DNA is conserved between species and it can't tell you which species those are. And so on to mass spectrometry. So mass spectrometry has been widely used in the analytical community. It's a great tool. It's very sensitive, selective. It's the tool that can quantify really low levels of something, and it's based on the mass of the compound you're looking at. And so everything's made up of amino acids. We can tell with very high resolution and actually figure out what amino acids there are in compounds that may be present as a contaminant or an unidentified component in a food product. And so my work started when I was diagnosed with celiac disease, looking at wheat proteins and trying to identify physiologically relevant peptides or parts of those proteins that would trigger the celiac autoimmune reaction. And there are lots of researchers out there now, they're doing the same thing, which is great because there's a real big and growing database of peptides now that we know are physiologically relevant. And so when I published this, we were just looking at wheat. We've since looked at barley and rye in the same fashion. And I'm going to be talking about how I'm using these peptides together in an assay where we can look at multiple different allergens in one run, which is very efficient and comprehensive. And so the beauty of LCMS is that there are no fudge factors. You're measuring the concentration of a peptide, which is directly proportional to the concentration of the allergen. And I'll explain how we do that. So here's what a high-resolution mass spectrometer looks like in my lab. I use proteomics. And what is proteomics? It's looking at protein. And so analytical chemists love to subdivide things and put an omic at the end of the name. And so in this case, proteomics of food analysis, some common food omics. People look at food analysis for the purposes of authenticity, food fraud, and food labeling. Allergenomics, people like myself, I guess, really focus on allergenic proteins for helping further medical research, especially with things like celiac disease. What proteins are allergenic? Also, other of the big 8 allergens that the US considers on their hit list. What proteins in, for example, walnut are allergenic? What peanut proteins are allergenic? So all those things fall under allergenomics. So you can look at proteins, we can say, from the top down or from the bottom up. And so the top down, we can look at the whole protein using mass spectrometry, or we can look and break it down into its components or peptides and look at those. And so it's basically what I'm showing here. What I do is take proteins, break them down in a certain way, and look at the peptides that are relevant to celiac disease. And so the acronym LCMS is liquid chromatography mass spectrometry, and that's what the instrument here is in my lab, one of the instruments. And its focus is really to look for trace amounts, really low levels. And so here's the workflow of an experiment. We start with extracting the protein from our sample, which is not trivial, very difficult. So we take the proteins, we break them up. Here's a relevant peptide that's a significant peptide here. This is a 33 amino acid sequence that was identified as one of the most toxic or physiologically relevant wheat peptides. And we run it through our instrument and we get something that looks like that. So the peak here is a very specific marker for this peptide, or any specific peptide you're looking for. And so we look at how big is this, how sensitive our method is to show these, and then we can use many, many, many different peptides in our method to study the food samples. Okay, so now I'm going to start throwing some data at you. And I just want to highlight some initial work that we did here. This is looking at wheat and looking at 7 of the maybe 25 to 35 wheat peptides that now that we have, looking at native wheat flour using flour from the US, Canada, and China in this experiment, and looking at fermented and hydrolyzed samples such as beer and bread. And we're looking at each one of these peptides and see how they respond in all these different samples, just to get an idea of if they're in every sample and how the sensitivity varies. And here again are the same 7 peptides, but we're looking at different strains, different species of wheat from around the world. Some are more common here in the US, and some are really more ancient or older varieties of wheat. And again, you can see how some of these peptides are much more sensitive than others. Sensitive meaning easier to analyze. And so we compared those same wheat samples using and rye and barley as well, with some of the commercially available ELISA kits that are used. And you can see how it's over 100 times different in some of the kits to some of the same analytical samples. And so that raises the question, why is that? Well, so this is one of the disadvantages of the ELISA test, is that every kit has different antibodies in it, and they respond differently to different proteins. And so that can be a problem in some types of samples. So our approach is looking at, as I mentioned, different peptides. Here are some barley peptides that we've identified, and you can see the different response in these. And our question is, well, how do you get from looking at one peptide to a concentration of gluten in a sample? And so the key is we developed a means to create a calibration with each peptide that we looked at and directly reflects the concentration of that target in the sample. And so sort of going around this circle of starting with a sample of protein, breaking it down, looking at the response, and then putting the picture back together to figure out what the concentration is. So another factor that we looked at for which peptides we should look at is the sensitivity, but also the specificity. Are they good peptides to look at in our analysis? This is a beer experiment we looked at, and we found that some of the more sensitive peptides may not survive the fermentation process. So they would not be relevant targets for us to look at in fermented or hydrolyzed products. And so you can see here, this particular peptide going through the fermentation process is chopped up versus this one over here, which remains pretty consistently well responder. So here's a calibration curve, what I mentioned. For each peptide, we have a peptide related to a direct matrix calibration, and we end up with a calibration curve which is linear over many orders of magnitude concentration. So we can look at a very low level of that, and we can look at a very high level. So here's an example of a low level. Here's a really low level of barley. That's what a peak looks like, relative to the standard which we use in our calibration. So now I'm going to show you some examples, just some quick examples. We do a lot of, everybody's interested in studying alcohol, so we looked at a lot of beer from all over the place, and my students in my lab love that because they get to drink all the beer after we're done. So here's an example of a low level of barley in beer, just what it looks like. This happens to be an ELISA result from a gluten-reduced beer that would be labeled gluten-free, but obviously it's not. Here's some examples of different peptides we use for barley, wheat, and rye, looking at some pretty hefty gluten concentrations there. And so these are as many as I could fit on this slide. Some really interesting, yeah, my students just love this experiment. And so I just want to draw your attention to these two, because these two you may know as gluten-reduced, or however they're calling themselves in whatever state you're in, I think. And then the apple orchard cider, and you can see they claim that their beer is less than 10 ppm, and that's what we saw also using our method, versus the Angry Orchard cider, which we know is inherently gluten-free. And so oats. Oats is very controversial, and everybody has, and I'm sorry, Marilyn, I know you didn't want me to talk about oats, but.
It's not that I don't want to talk about oats, it's that you have 2 minutes to finish.
Okay. So we were asked to look at this, and there was some questionable ELISA data from this, and so it turned out that there wasn't wheat in the oats of this particular sample, but there was barley. And we looked at a lot of commercial foods between some ELISA companies, and you can see the discrepancy that we're getting, which is why mass spec can help in a lot of cases. We looked at spice contamination. So ELISA can fail sometimes because of the colored samples in spice. And so our method here, we're looking at all kinds of different allergens, as well as wheat, barley, and rye. So you can see in an unlabeled product, doesn't contain tree nuts and so on. Well, there are lots of allergens in there. You can see here various different sources of spices with wheat, barley, and rye, and some scary amounts that aren't labeled. Dietary supplements, we looked at, they're not really well regulated, but we looked at probiotics with the group at Columbia. Found several were labeled gluten-free, several weren't labeled gluten-free, several contained gluten, which is scary and very upsetting to the GIs that prescribe probiotics for newly diagnosed celiacs. A lot of commercial food, we did a study with Columbia and found, thankfully, most of gluten-free food is, in fact, gluten-free. And so I guess I want to, leave this talk with, I hope you all have a better appreciation for how difficult this type of analysis is, and it's not trivial, the work that people are doing in this area to make the methods better for all of us around the world to eat safely. We have a lot of vested interest in making a mass spectrometry assay that's accredited by the FDA to help in manufacturing arenas to make sure that any problems, there's a second orthogonal technique that can be used to help. And my most important takeaway message to everybody is read the labels of what you're eating. And I'm going to say the same thing Janelle did. Ask questions. Don't assume that what you're eating is gluten-free if it has a label. Just be careful, and please go and see your GI doc and your nutritionalist regularly. And I'd like to thank all these people for help and sending me samples and helping with all this method development over these last several years. And thanks so much for your attention.